welcome to this deep dive. We're going to be exploring alternative models in muscle development and inherited myopathies research.
Oh, very cool.
Our focus today is a comprehensive review from BMC Veterinary Research.
Right.
It's called Aligning with the 3 Rs, Alternative Models for Research into Muscle Development and Inherited Myopathies.
Very interesting.
So,
Yeah.
it's a very timely topic, as the field is increasingly focused on replacing, reducing, and refining the use of mammalian models in research.
Right. And, this article really emphasizes the critical role of skeletal muscle, in overall health and movement.
Yeah, and, I think we often overlook just how essential these tissues are.
Yeah.
They constitute a significant portion of our body mass and are major energy consumers.
Precisely. And when you consider inherited myopathies like muscular dystrophies and congenital myopathies, the impact on both human and animal health is profound.
Yeah.
These disorders can cause significant morbidity and mortality, making the search for effective treatments, even more urgent.
Absolutely. And the review does a great job outlining the limitations of traditional mammalian models.
Right.
Not just from an ethical perspective, but also considering cost regulatory hurdles and the difficulty of studying early developmental stages in utero.
That's why the 3 Rs framework is so crucial.
Yes.
This ethical framework focusing on replacement, reduction, and refinement offers a roadmap for more humane and efficient research practices. The emphasis on replacement is particularly intriguing as it explores ways to completely or partially replace animal models with less sentient alternatives.
Right. So the article delves into several promising alternatives, starting with in vitro models, often referred to as muscle in a dish.
Right.
This approach utilizes cultured myoblasts, which can be derived from established cell lines like the C2C12 line from mice.
Yeah, C2C12 cells are readily available and offer an ethical advantage over using live animals.
Yes.
However, it's important to acknowledge the limitations.
Right.
While primary myoblast cultures closely resemble native muscle tissue, they have a limited lifespan and are challenging to maintain. Immortalized cell lines like C2C12 can proliferate indefinitely, but may experience genetic drift over time,
Yeah.
deviating from their original characteristics.
Okay, so then the article kind of transitions to discussing the evolution from traditional 2D cell cultures to more sophisticated 3D environments.
Right. This is a significant advancement. 2D cultures, while convenient for imaging and genetic manipulation, lack the structural complexity of mature muscle tissue.
Right.
3D cultures aim to bridge this gap by providing an environment that more accurately mimics in situ conditions.
Okay, yeah, so the review highlights several types of 3D models.
Right.
Could you elaborate on those a bit further?
Certainly, scaffold-based methods are one approach where cells are cultured on artificial matrices composed of materials like collagen or matrigol.
Okay.
These scaffolds provide structural support and allow cells to interact in a more physiologically relevant manner.
Got it.
Another approach is the use of organoids.
Okay.
These are self-organizing clusters of cells that resemble the target organ, in this case muscle tissue. Organoids offer a higher level of complexity, recapitulating aspects of tissue architecture and function.
So the review also mentions organ-on-chips, or OOCs,
Right.
which seem particularly innovative.
OOCs are microfluidic systems that recreate organ-level physiology on a chip. Imagine a tiny device with channels and chambers that mimic the flow of nutrients and fluids, just like in a real muscle. These systems allow for real-time monitoring of cellular behavior and responses to various stimuli.
While these in vitro models offer significant advantages, the review acknowledges that they cannot fully replicate the complexity of a living organism.
Right.
So that brings us to the realm of computational models,
Okay.
which leverage increasing computing power and data availability to simulate muscle function and disease progression.
Computational models, or in silico models, utilize mathematical equations and algorithms to represent biological processes. For instance, they can model the mechanical aspects of muscle contraction, predict disease progression based on known factors, or evaluate the effects of potential therapeutic interventions.
And the review cites examples of computational models being used in Duchenne muscular dystrophy, DMD, research,
Right.
highlighting their potential to accelerate drug discovery and personalized medicine.
However, it's crucial to remember that these models rely heavily on the accuracy and completeness of the data input.
Yeah.
If our understanding of muscle biology is flawed or incomplete, the model's predictions will be unreliable. It's a constant process of refinement and validation.
So we have these powerful in vitro and in silico models, each with its strengths and limitations.
Yes.
But what about alternatives that offer the complexity of a living organism without the ethical concerns associated with mammals?
Right.
The review explores the use of invertebrates,
Okay.
specifically Drosophila melanogaster, fruit flies, and Caenorhabditis elegans, nematode worms.
While seemingly distant from human biology, these tiny creatures offer surprising advantages for muscle research.
Could you elaborate on their relevance to understanding human muscle biology?
Fruit flies, for instance, possess multinucleate muscle fibers, a feature shared with human muscles. They have been successfully used to model aspects of muscle development, aging, and even human muscular dystrophies.
Wow.
C. elegans, with its fully sequenced genome and well-studied genetics, provides valuable insights into sarcomere assembly and muscle function.
Oh.
Despite their structural differences, they have been instrumental in modeling DMD and screening potential drugs.
The review highlights the ethical and practical benefits of using invertebrates,
Right.
such as their ease of maintenance, rapid reproduction, short lifespans, and well-defined genomes.
These advantages make them ideal for large-scale, high-throughput screening, allowing researchers to test thousands of potential drugs or genetic modifications in a relatively short time frame.
But the review does acknowledge that invertebrates are not a perfect replacement for mammals.
Right.
Their muscle structure and physiology differ in key ways, and they lack certain mammalian systems relevant to complex diseases.
That's precisely why it's important to choose the right model for the specific research question, carefully considering the strengths and limitations of each approach.
And that leads us to another fascinating model discussed in the review: embryonic zebrafish.
Okay.
While adult zebrafish are vertebrates, their embryos offer a unique window into muscle development, bridging the gap between simple invertebrates and complex mammals.
Very cool.
What makes them so well suited for muscle research?
Their embryos develop externally and are transparent, allowing for real-time observation of muscle formation without invasive procedures.
Interesting.
They also develop rapidly, with muscles forming within days, making it possible to study the entire process in a compressed time frame.
Oh, okay.
And like invertebrates, they're amenable to genetic manipulation, enabling the creation of models for various muscle diseases.
The transparency of zebrafish embryos is particularly intriguing.
Yeah.
Did the review mention any specific insights gained from this unique feature?
Absolutely. Researchers have used this transparency to track the precise timing of gene expression during muscle fiber formation. This has provided valuable insights into the molecular mechanisms underlying muscle development and the causes of certain congenital myopathies.
The review also highlights the use of zebrafish embryos in high-throughput drug screening.
Right.
It seems like they offer a powerful combination of ethical considerations, vertebrate-specific features, and experimental advantages.
Yeah.
But are there any areas where they fall short?
While zebrafish embryos offer a valuable model system, it's important to remember that their muscle structure and function still differ from mammals in some ways.
Okay.
Their locomotion is primarily driven by their tails, and the forces acting on their muscles are different from those experienced by land-dwelling mammals.
So it seems like each model, from cells in a dish to swimming zebrafish embryos, offers a unique perspective on muscle biology.
Right.
The key is to understand the strengths and limitations of each approach.
Yeah.
And choose the most appropriate tool for the research question at hand.
Exactly. And that's where the 3Rs framework becomes so essential.
Yes.
It guides us towards a more ethical and responsible approach to muscle research, ensuring that our pursuit of knowledge minimizes harm and maximizes the value we gain from each model.
It's about recognizing that each model is a piece of the puzzle.
Yes.
And by combining those pieces, we can gain a more comprehensive understanding of muscle biology and disease.
Precisely. The more diverse our toolkit of models, the better equipped we are to unravel the complexities of muscle function and dysfunction. It's about recognizing that each model is a piece of the puzzle, and by combining those pieces, we can gain a more comprehensive understanding of muscle biology and disease.
Yeah, and this integrative approach seems particularly relevant as we move towards personalized medicine,
Right.
tailoring treatments to an individual's specific genetic and environmental factors.
Precisely. The more diverse our toolkit of models, the better equipped we are to unravel the complexities of muscle function and dysfunction.
And the review also emphasizes the importance of the 3Rs in guiding the development and application of these alternative models.
Absolutely. As we strive to reduce our reliance on mammalian models, it's imperative to ensure that our research practices remain ethical and humane.
Yeah, the review cites several examples where alternative models have led to significant breakthroughs in muscle research.
One notable example is the use of zebrafish embryos to screen for potential drugs for Duchenne muscular dystrophy.
Oh, okay.
These screens have identified promising candidates, including phosphodiesterase inhibitors and even fluoxetine and antidepressants.
Oh, wow. It's fascinating how research in one area can unexpectedly yield insights into seemingly unrelated fields.
It highlights the interconnectedness of biological systems and the value of exploring diverse approaches. A drug initially intended for a mental health condition might hold the key to treating a debilitating muscle disease.
Yeah.
That's the beauty of scientific discovery.
The ultimate goal of all this research, of course, is to develop effective treatments and ideally cures for these devastating muscle diseases.
Are we getting closer to that goal? That's the crux of the matter. While we don't have all the answers yet, the progress made with alternative models is certainly encouraging. We're gaining a deeper understanding of the underlying mechanisms of muscle diseases, identifying potential therapeutic targets, and developing more efficient and ethical ways to screen for potential drugs.
It's a dynamic and rapidly evolving field, and it's exciting to witness the progress being made.
The convergence of disciplines from cell biology to bioengineering to computational modeling is driving innovation and creating new possibilities. The future of muscle research is bright, and the 3Rs will continue to guide our path, ensuring that our pursuit of knowledge aligns with our ethical responsibilities.
The review does a great job of highlighting not just the scientific advancements, but also the ethical considerations that must guide this research.
It's a reminder that scientific progress cannot come at the expense of animal welfare. The 3Rs provide a framework for balancing our quest for knowledge with our responsibility to minimize harm.
Yeah, the review emphasizes that no single model is perfect. Each has its limitations, and it's essential to understand those limitations when interpreting research findings and drawing conclusions.
That's a crucial point. We cannot simply extrapolate findings from one model to another or assume that what works in a dish will necessarily translate to a living organism. It's about using different models to complement each other, to build a more complete picture of muscle biology and disease.
And the review also highlights the importance of transparency in research, ensuring that methods and findings are clearly communicated, allowing for critical evaluation and replication.
Transparency is essential for building trust in the scientific process and ensuring that research findings are robust and reliable. It fosters collaboration and allows the scientific community to collectively advance our understanding of muscle biology and disease.
This brings us to a broader question. What is the overall takeaway for our listeners? What does this exploration of alternative models in muscle research tell us about the future of scientific inquiry?
I think it points to a paradigm shift, where ethical considerations are no longer seen as separate from, but rather integral to scientific progress. We're moving towards a more holistic approach, where the pursuit of knowledge is guided by compassion and responsibility.
It's inspiring to see how researchers are embracing this shift, finding innovative ways to study complex biological processes while minimizing harm and maximizing the value we gain from each model.
It's a testament to human ingenuity and our commitment to advancing knowledge for the betterment of humankind and the animal kingdom alike.
The review concludes by emphasizing that the ultimate goal of all this research is to improve lives.
Whether it's developing new treatments, improving diagnostics, or simply expanding our understanding of how our bodies work, it's all driven by a desire to make a positive impact on the world.
And with these alternative models paving the way for more ethical and efficient research, we can be optimistic about the future of muscle research and its potential to alleviate the suffering caused by these debilitating diseases.
I share your optimism. And as we continue to delve deeper into the mysteries of muscle biology, let's remember the lessons learned from these alternative models, the power of innovation, the importance of ethical considerations, and the unwavering pursuit of knowledge that benefits both humanity and the animal kingdom.
Yeah, it's a powerful reminder that science is not just about cold, hard facts, but also about compassion and responsibility.
Indeed, as we push the boundaries of scientific knowledge, it's essential to consider the ethical implications of our methods and strive to minimize harm whenever possible.
This review from BMC Veterinary Research serves as a valuable resource for researchers and anyone interested in the future of muscle research.
It provides a comprehensive overview of alternative models, highlighting their strengths and limitations, and emphasizes the importance of the 3Rs framework in guiding ethical and responsible research practices.
It's a call to action for the scientific community to embrace these alternative models, to refine our methods, and to ensure that our pursuit of knowledge aligns with our values.
And as we conclude this deep dive, let's remember that the ultimate goal of all this research is to alleviate suffering and improve the lives of those affected by muscle diseases. It's a challenging but ultimately rewarding endeavor, and the progress being made with alternative models gives us reason to be hopeful.
From Vet Neurojar, keep those minds-inspired hearts light and tails wagging.